Good evening, everyone. Let's start our fourth public lecture of the year 2008. It's a great pleasure for me and also a big honor to introduce my colleague and friend, Vittal Yachandra. Vittal was born in India and he got his PhD at Princeton University in 1982. Since then, he's working at the Lawrence-Berkeley National Laboratory, and he has, since that time, been specialized on studying the light cycle of photosynthesis. Vittal has been one of the most active users of the Stanford Synchrotron Radiation Lab, which is down the hill. In fact, he's coming here since 1977, since over 30 years. And if I would have to count in terms of how many hours he really has spent here, I would easily rank him in the top 10 of any user we ever had. Probably higher up. Vittal is also an expert on cricket. And because the most interesting matches always take place during the night, he always takes the night shift during beam time. Today he will guide us on an incredible journey, starting at the dawn of evolution and maybe leading to the future of our planet. Please join me to welcome Vittal Yachandra and the talk, How Plants Do It, Light, Oxygen, Action.
Thank you very much, Uwe. Can you all hear me well? Okay. Just bear with me for a few minutes with my Indian accent. You'll get used to it in a couple of minutes. I'd rather be playing cricket. Giving a talk is a hard thing, preparing for it. And in fact, India is playing Sri Lanka, I think, right now, this evening. So it's a good thing to go and watch. It's wonderful that all of you come to such talks, and I'm from the other side of the bay, the little college out there called UC Berkeley, and I wish we could do that there. I think this is fantastic to have such an audience come up for talks like this, and for us to try to make things, it's difficult to sometimes talk in a way which makes more interesting than we normally do in seminars. So it's a challenge. So if it is a little bit confusing or whatever, bear with me. Eventually the story will come through. Today's story, I'm going to tell it to you in 3 parts. The first part is, we all breathe oxygen, there it is. But did we ever wonder where it comes from? Or how do we have so much of it? It's a reactive gas, and there is so much of it. How did it accumulate to such a level? And second part of my story, I'm going to tell you about how plants actually produce this oxygen. That's obvious. That's what the title is. And they produce it from water. I'm going to tell you about this miraculous engine which does this reaction, and how and why it is important, and all the methods we're using to figure out what the structure is of this engine. It's a huge engine in terms of a molecule, which is very tiny, but it's a fascinating story. How oxygen collected in the atmosphere, how this engine made this possible. And we thought that was the end of the story when I started this research in 1980s, early 80s. And we were writing our grant applications. We're going to save the world. Please give us money. Because understanding the reaction is really, really important for so-and-so reason. And it was always with a little bit of a smirk. But right now, the smirk is completely gone. It is really true. And that's how my story is going to end. That's the third part of my story. And let me start with the first part. Where did all this oxygen come from, and how did it all begin? Okay. In the very beginning, there was the Hadean. in terms of our paleontology. The Hadean means the hell. Basically, our Earth looked like hell, at least artist's conception of hell. Here you see volcanoes spewing all kinds of noxious gases, mostly sulfur and methane and whatnot. We had a very, very high concentration of CO2. We'll come back to that in the last part of my talk. This is going to be very important, what is happening to us right now in terms of our environment and our energy problems. And there was also already by 4 billion years, there was enough water. Before that, Earth was impossible. But by 4 billion, it had cooled enough. There was enough water. And underneath the surface of the sea level, somehow, miraculously, life started. We don't know how it started. After it went on merrily along for 500 million years, life went on underneath the sea, and mostly they were tiny little bacterium, which kept going on. They were feeding off sulfur, nitrates, all kinds of things which were in the sea. And by the way, in the sea, we had these black smokers. I couldn't get this thing to animate, but it's pretty fantastic. Underneath, even right now they exist. They just spew out all kinds of noxious things, mostly rotten egg smelling hydrogen sulfide, which, by the way, the bacteria like. They use this as food. But, I shouldn't say but, there were also lots of minerals coming out, iron and manganese. These are very common minerals on the Earth's surface, and that will become important later on too. And somehow these bugs, we'll call them bugs, the bacteria, though it's not technically right, and they somehow started also at this point using light. They used light to make energy and also feed off the sulfur and nitrates in the bottom of the ocean, or wherever in the ocean. I shouldn't say bottom, probably in the lower levels. So there were bacteria, and there were bacteria which were using light. But after about 500 million years or so, the bacteria started depleting the sulfur and the nitrates, the food sources. Okay? And there was one smart bug which looked around, saw a lot of water, and said, I'm going to use water as my food source. I'm simplifying it, but that's exactly what it did. Please forgive me. It didn't do it consciously, but that's what happened. It started using water as a food source. And something amazing happened. When you use water as a food source, it started excreting oxygen. Can you believe this? Oxygen was a waste product for this bacteria. So, unintentionally, oxygen was coming out of the oceans. So, it is the excretory product of the bacteria, of these bacteria which had learned to use water as a food source. Okay, what did the atmosphere look like at this point? It had the most foul-smelling stuff, methane, H2S, which is rotten egg, ammonia, water vapor, and a huge amount of CO2, carbon dioxide, and all at very, very high pressures. If we are at 1 atmospheric pressure, now it is 10 atmospheres. And there was a lot of minerals here. So that was the state of affairs, and these bugs had now learned how to use water. I'm going to call it splitting water. It's not like splitting atom. When you split a molecule, you get atoms, smaller molecules. So I'll call it splitting water. So when you split water, these bugs, which are called cyanobacterium, because they are kind of blue-green in color, so they're called cyanobacterium. Look at it. This is now in billions of years, and this is the amount of oxygen collecting in the atmosphere. This is we have 20% oxygen right now. So there was very, very tiny amounts of oxygen, all in the sea, because all life was still under the sea, because life was impossible above the surface of the water, because there was too much ultraviolet radiation. We know what ultraviolet radiation does to us. And the same thing to bugs. It kills them in large amounts. And there was a huge amount of ultraviolet radiation, Because there was no ozone layer. Because there was no oxygen, there was no ozone layer. So there's a huge amount of UV light coming in, killing all the bugs. All this life was underneath the water. And most of the bugs were still using sulfur and nitrate. And these guys started evolving oxygen. They poisoned the entire thing. This was the first large-scale environmental disaster. The poisoning of the earth with oxygen. Can you believe that? Now we take it for granted that we all breathe and life depends on it. But actually, it actually poisoned the atmosphere 3 billion years ago. It's kind of hard to imagine what 3 billion years is, but we'll get to it. So it started collecting slowly, very slowly. This scale is not linear. It goes in powers of 10. So it took a heck of a long time for it to start accumulating. The reason being, it was being used up. Okay, first was, I forgot to tell you, that is, this is an actual picture of the cyanobacteria, which first started to learn to split water. And that is absolute milestone in evolution, because without these things, we would not be here today. The second step, of course, oxygen started collecting, and there was a lot of iron in the seas, and they started precipitating. All the iron got oxidized with oxygen and fell down to the bottom of the ocean. Many of these things are controversial. There is a lot of opinions, a lot of things. I'm generalizing it. So for a long time, it kept doing that. And if you go to the bottom of the ocean, you see these things. And some places where the oceans have been exposed, you see these beautiful iron bands. That's why it's called BIF, banded iron formations. So after all the iron was depleted, other things were also getting oxidized. It started escaping into the atmosphere more and more. Now it is at 0.1% here. And it took almost 1 billion years before this one cell, all these bacteria are one cells. And they don't have a nucleus. Then you started getting what are called eukaryotes. Eukaryotes are basically things which have a nucleus in the cell. So things like these bacteria entered into other cells and became, it's called symbiosis. It's called endosymbiosis in this case. So they formed a more complicated organism. So for 1 billion years, that's all they were. These are the only things which were doing oxygen evolution. So you got more complex lives after 1 billion. And still nothing happened for a long time. Again, look, this 1% oxygen, still life was very, very simple because the things were not respiring. There was oxygen, but unless you burn oxygen to make fuel, you can't get more complicated forms of life. And that's what happened about 500 billion years ago. Sorry, 500 million years ago. It's called the Cambrian explosion, where everything we know, all the life forms we know, every vertebrate system, mammals, the pre-mammals, everything started evolving there. And at about that time is where we also got land plants, because there was enough ozone in the atmosphere that they could crawl out onto land. There was enough ozone to protect the UV light coming in. So plants could come out into the land. And that's what I have here. And that only happened 400 million years ago here. This is where life started somewhere here, and only plants came up about here. That's pretty amazing to me. We think about plants as being there forever, for a long time, but it's not true. Land plants came only about 400 million years. Okay, the story now is, we had a really noxious atmosphere then, which is 4 billion years ago, and now, what do we have? CO2 has decreased considerably. From 50%, it has decreased to 0.04%. That is an amazing decrease in carbon dioxide. And look at now what we have. Mostly nitrogen and oxygen. The little sliver is all the other gases. And most of this oxygen is the work of these little tiny one-cell cyanobacteria. What the plants contributed is not that much in the overall evolution of life. Right now they contribute about 50%, and the cyanobacteria still contribute about 50%. Okay, let's put them all together in this evolution clock. It's kind of a fun thing to look at it in a clock like this. This is 5 billion years cycle. So origin of Earth is about there, 4.7 billion years. And then life started somewhere around here. It's controversial, 4.2, whatever, somewhere around here. And then if you look at it in this scale, you see the rhythm of life here, where cyanobacteria started. It's about here, 3.2 billion. This is what started poisoning our atmosphere. And terrestrial plants didn't come up till about here. So all this stuff was going on. And this is where the Cambrian explosion. Vascular plants means which pump fluids up and down. There are actually veins and arteries in plants like that. And most of them were ferns, and I'm not much of a gardener, but all these plants don't have seeds and don't have flowers. And then, of course, the dinosaurs came about 250 million years ago and disappeared by about 65 million years ago, a very well-known fact. And modern humans about 250. It's just my tiny, tiny sliver. The important thing for this talk is that plants didn't come up onto the land till about 400 million, and flowering plants didn't come up till maybe the time the dinosaurs were disappearing. So interesting fact that dinosaurs probably never saw a magnolia or a rose. We see these beautiful pictures of dinosaurs roaming around in these gardens. They should all be ferns. In that case, it's pretty good. Or trees which don't have, which are called angiosperms. And to me, this is interesting. We act like we are the center of the universe. We are a tiny, tiny part there. I already told you this. This is a fascinating story. These cyanobacteria, which poisoned our atmosphere, I like saying that because it's such an interesting fact. They still exist in fossil form, and these fossils have been dated to almost 3 billion years ago in Western Australia. What they do is they form mats. And this picture tells you what they do right now. They exist. This is a living cyanobacterial mound. They die and they form more on top, and they die and form more on top, and you get these in fossils. And this is amazing. The fossils of that age exist, and they still thrive. And the interesting thing, which I'll come to later, is the machinery which makes the oxygen is the same in these guys and in those higher plants. So they're separated by 3 billion years of evolution, and the same thing works in both of them. And we've shown that with our experiments. Okay, that is the first part of the story. These bugs, critters, poisoned our atmosphere with oxygen, made life possible for people like us, higher forms of life, all starting around 500 million years ago, and more complex forms of life. And how did this happen? I told you already. They were using light, and they learned to split water. So this is what we learned in, this is the second part of my story. I'll tell you about how the engine works, okay? We finished the second part. This is a textbook example of photosynthesis. Photosynthesis we talk of as, everybody knows what it is, there's nothing to know about it. But what I just told you, how oxygen is formed from water, we really don't know how still it does it. So keep that in mind. When we say we know everything about photosynthesis, we're talking about something else. It's a complicated process where we use light, the sun, carbon dioxide, which used to be in a large amount, now it is very tiny, and it's increasing again. Water, which is in plentiful supply. And then we make sugars and oxygen. This is an interesting thing. Let's go through it slowly. This is, by California standards, a very average tree. But Uwe tells me it is a German tree, and it is a very special tree. It's some kind of a Bavarian birch or something like that. a tree like this produces 10,000 liters per day of oxygen and 12 kilograms a day of sugar. And this is spinach, which is what we use for our experiments. So we go to the market, buy spinach, grind it, and make our samples from it. And these are the cyanobacteria which I told you about earlier on. So spinach is where all the information we know about this, most of the information we know about it comes. Okay, when we say a tree evolves 10,000 liters of oxygen a day, it's very hard to really know it's actually doing it because you don't see oxygen coming out. But if you ever if you ever had an underwater plants, unfortunately I couldn't again animate this thing, you can actually see these oxygen bubbles coming out. It's fascinating. It's like one of our colleagues called it the champagne of reactions. And remember, what I just told you about photosynthesis, it's a complicated process. It consists of two different parts, which I'll tell you about right now. So, you use sunlight, you use carbon dioxide, you make starch, and then you evolve oxygen. Where does this oxygen come from? How does these plants do that? That is our research. That is our research program. And I'm going to spend the next third of my talk telling you what we know about the structure and what places like SLAC and SSRL make this possible. It's studying spinach, but trying to understand life in its most basic forms here. And what we need to do that is these complicated, expensive instruments here. And I'll tell you in as simple way as possible what it is. Okay. This, obviously, photosynthesis occurs in the leaves. You take a section of the leaf, you see these beautiful pigments which absorb the light, which are called light harvesting pigments. And they gather the light and send it to the business end of the leaf, where the reactions take place. Just going further down, take one of these things and look through the small sections, you see what are called chloroplasts. This is a word which you probably have come across. The chloroplasts are really the ancient cyanobacteria which got themselves into the higher forms of life. This is what I call symbiosis. These guys look very much like a tiny cyanobacterium. And you go in even further, you see these beautiful membranes. Membranes are basically lipids, fat molecules, which are arranged in parallel form, so they form a little nice flat layers. And all the things which happen in photosynthesis are embedded in this layer. This is a membrane. So you see the two-dimensional membrane? And in the membrane are all these proteins. And this is where all the photosynthesis goes on. The simplest form which we have known for a long time is how do you get starch from carbon dioxide. And that is called the Calvin cycle. This is something, again, we learned in high school. And I'm very proud to say we are from the Calvin lab in Berkeley. And Calvin, before I go to that, let me just tell you, this consists of two parts, photosystem 2, photosystem 1. Photosystem 1 is where starch is made. I shouldn't say exactly in photosystem 1. It is more directly connected to the making of starch, while in photosystem 2 is where the splitting of water occurs. This is done by a catalyst, a manganese-containing cluster. Remember I told you in the early part that these black smokers were throwing up manganese and iron? These bugs picked it up. And they made a catalyst from it, which makes this reaction possible. Okay. Sorry. So using all the techniques which were available in the 60s, early 50s, late 50s, Calvin figured out how starch is made by plants. So when we say we know all about photosynthesis, that's what we're talking about. We don't know anything about what happens here. I shouldn't say anything. We don't know much less about it. And for that, Kelvin received the Nobel Prize in 1961, and a laboratory in Berkeley was named after him called the Kelvin Laboratory. It was already futuristic. It was built in 1966. Already was thinking about different people working together. And when I came to Berkeley in 1982, I was part of this building. He was still alive and active those days. And he was very much interested in the other half of the reaction. How does that happen? And now it has been dedicated to biofuels research, and we had to move to a different building last year. Okay. I'm going to tell you about now, this is the technical part of my talk, where I'm going to try to tell you about this molecular engine and how it is put together in the most basic ways and how it splits water. We talked about photosystem 2 here, remember? We talked about this complex. This complex is a huge number of proteins which are put together in a very, very specific way so that this particular reaction can occur. The reaction is take water and make oxygen. It's hot here. It's hard work. Excuse me. Well, okay, this is a cartoon again of photosystem 2, where you have the leaf, we have the membranes, and we have the proteins, and we have all the pigments which absorb the light. Once the light is absorbed, this particular molecular machinery splits water to oxygen, and in the process produces hydrogen, okay, or the equivalents of hydrogen in a simplified way, and it is catalyzed by this complex. When you say in chemistry catalyzed, what do we mean? What we mean is that it makes a reaction possible, it makes it go, it makes it easier. Without that, you would not be able to do it. So these bugs somehow figured out to extract this manganese, which is in small quantities is enough to make this complex. What do we know about this complex? And this is where SLAC comes in, SLAC and SSRL. They have the most amazingly beautiful instruments. And the very first time I came to SSRL as a graduate student in 1977, I absolutely fell in love with it. I was hooked by the amount of instruments that are available, the kind of facilities, the kind of research you can do. And specifically to this problem which I'm talking about, we have this huge, humongous biological system. You have this tiny little manganese complex there, and you can study it using the facilities we have here. And how do we study it? We have this is called the electromagnetic spectrum. All the forms of light. You have all the wavelength of light starting from a huge radio waves all the way up to the kind of wavelengths we use for our studies, which is in the X-ray region, which is what makes SSRL famous. It produces some of the nicest X-rays, and they also produced it much before anybody else anywhere in the world could do that. And we use the X-rays, which is a very short wavelength, very high energy X-rays, to study the structure of these biological molecules in a very, very specific way. And in a very, very, what shall we say, the atom we want to study, not the whole thing, where the action is. How do we do that? Well, we all know this one, the famous periodic table of all the elements. And if you take our spinach squash, what should we call it? Spinach smoothie, okay? Which is what we do. We have a big blender, $800 blender. Can you imagine a blender costing $800? I don't know why it costs, because it's just like the $50,000 toilet seat. It's just no reason for it to be $800. Anyway, that aside, we blend the spinach in this blender, and what we have in spinach at that point mostly are these elements which are shown in green. And remember, we are manganese and iron. Again, we come back to them. And using the X-rays available here, which are very intense, and you can choose what you want. So you can choose that, and you can study the spectrum of just that element. Okay? You have literally tens of thousands of atoms in your molecule, and there are only 4 manganese atoms in this complex which do this reaction, and we can study those 4 atoms using the X-rays. And the reason is you can separate out the spectrum of each element. Manganese, iron, carbon, oxygen, everything is spread out. You can study manganese alone. Remember, this looks like nothing. You know, it just looks like some wiggles, but these are extremely important wiggles. To get that took us 10 years more, and I'll tell you why. Or I won't tell you why. I'll tell you what we can get from those wiggles. A little bit of what actually you can get from these wiggles. This is a beautiful animation made by my friend and colleague Uwe Bergman. You have the atom. Inside the atom, you have a nucleus. You have a whole bunch of electrons in different orbits. It really are orbitals, they call, but for simplicity, orbits. So you can, if this is manganese, let's call it, then you can find out about the manganese. It's called the structure of the manganese atom inside the atom. But what we are interested in is not just that, also what is around the atom, in the immediate surrounding, because we want to figure out how these things are put together in order to figure out how they work, just these manganese atoms. So how does it happen? How does it work? You shoot this atom with X-rays at SSRL, and then you get a wave of electrons going out, which meet atoms next door to it, and they come back. So they either come back in phase, like you drop 2 stones in a puddle, the waves either are constructively building each other or destroying each other. But intuitively it is obvious. The 2 stones, the distance between them determines whether it is constructive or destructive. So the mathematics of it is you can figure out how far they are if you know these things, which is what those wiggles I showed you about are. So you get these wiggles, and then you can figure out how far from manganese atom or other atoms. And once you know how far, you are pretty much talking about how this thing is built. The fun thing about doing these experiments is you get to travel around the world. And we really do travel. And I'll tell you about that in a minute, how our carbon footprint is so high because of that. Our favorite is, of course, SSRL, it's just second home for us, coming from Berkeley. You can go to Argonne, you can go to our own place in Berkeley, but it's a different energy region, which is also very, very good. Or you can go to Japan, and we've gone to all these places. And as I said, it is a lot of fun doing this. We collect all our equipment, put them into this huge, beautiful fiberglass boxes, which Department of Energy lets us buy, ship them to these places, and do the experiments, and then take 1 week off and go touring. So I managed to see much of Europe, Japan, all other places. But we come back here because there are things here which are perfectly tuned to biological spectroscopy, which we don't find in most other places. There are specific things you can do other places better, but here it is just a lot of other things here which are good. So I already told you that we can specifically look at manganese atoms, and what we're trying to figure out is how are these manganese atoms or this catalyst put together. For a chemist, that's a big deal, because once you know what the structure of a complex is, you can figure out how it works. Remember the reaction it is doing, splitting water? It's a fantastic reaction. You cannot do it in a beaker yet efficiently. And once you figure out how to do it, there are consequences which we'll come to before the end of my talk, which are extremely important for the present day. So we don't know what the structure is yet, but we made a heck of a lot of headway. And how do we do it? It's a real detective story. You get a little data, you proceed a little farther, little more data, you say, oh, that's wrong, go in another direction. And it is that is the fun of doing science, because every day is different. Every day you're plotting and planning, and in a way you're being devious with the system. How do you defeat it? How do you overcome it? And in our case, I'll show you how simple it is. It's got 4 manganese atoms. How many different ways can you put these 4 together? And the first thing we did, after more than 6-7 years of research using X-rays, we figured out this particular way is one very good way. Okay? That does a lot. It has to be manganese, it has to be 2 oxygens. So we already taken care of 2 manganese atoms. We need 2 more. We also figured out this particular unit also exists. So many people who've done a little bit of chemistry know each one of them is a different structure. But you have to have this, you have to have this. Think about it as a puzzle now. This has 2 of them already, and this doesn't have any of that. So you get the idea. This has to be there, and this has to be there. And 10 more years later, we figured out this has to be there. Calcium, oxygen, and manganese. And in order to get to this, there's an enormous amount of experimentation, enormous amount of data collection, all those wiggles I talked about. And then you distill it, and you come up with a whole library of structures. And then you figure out which works and which doesn't. So this is what I was talking about. Even after you know those 3 different ways of putting those atoms is there, you can put them together in a huge number of ways. Let's take, for example, this guy. Okay? It has this, which I talked about, this structure. It has this structure. It also has this structure. These are the 2 things I talked about. So, and there are only 4 manganese. The red is manganese, blue is oxygen. So that is one structure. Let's take something else, this one. That also has 2 and 1 like that. So a whole bunch of them. And believe me, it took us all the way through the 90s to come up with all these things, and none of them were still right. Because technology was improving, the experiments were getting more complicated, and more information was possible. We could extract more information from our experiments. And in late 90s, we figured out there had to be 3 of these guys. Now it's a question, again, puzzles. If you had 3 of them, and you had to have at least one of them, and you had only 4 of these things to put them together, how many ways can you do it? So if you sit around a table and with a pencil and paper and draw them, you'll get a lot of them. But there are also experiments to prove and disprove what all these things are. So all the things on this side of the line now have 3 of these guys. Let me pick one. Okay, let's pick this guy. You can see 1, 2, 3, and we need one of this guy, which is here, or there's one more here. So you get the picture. So we are playing these games. Okay, it couldn't be that. It can be this, it can be that. But for a variety of reasons, which we will not go into, we were able to reduce it to these guys, all in this blue box. And that is an amazing thing, from this whole huge collection of things, to be able to say that this catalyst, which does this remarkable reaction, has a structure something like this. And that has made us very well known in the world for that. I can't believe it. Just for these dots and these little structures. That's not the end of the story, because we've gone further than that. That's 2000. So it's taken us almost 20 years to do that stuff, to get there. Most recently, this is around 2004, me and my colleagues, many of them are here, came up with a completely new way of doing this experiment. Normally what you do is you take the spinach smoothie, as I told you about, we study that, and all the molecules are randomly there arranged. So you get them in every which way possible. And the information content from this is quite large, but it's very hard to rule out certain possibilities because of that. But what happens if you take a crystal? Can you believe these things which come from the membrane, from the biological system and spinach? You can actually crystallize these molecules, which are so big. You can get a crystal which is beautiful green color. This is only 0.5 millimeter, the scale. It's a tiny, tiny crystal. And now you do the same experiments which we did with the random samples. The molecules will all arrange in nice, regular pattern. Now what happens is something, you can reduce the number of possibilities very, very easily. I shouldn't say very, very easily, but you can do that. So voila. We figured out what it was to a very high degree of certainty. The red ones are the manganese atoms, the blue ones are the oxygen atoms, the green one is the calcium atom, and now we also have a pretty good idea of how it sits in the protein. It's not floating in the protein. It is sitting there nicely bound to the protein in order to do this chemistry. So it is a biological catalyst which has not been, what shall I say, reproduced by man yet. We have not figured out how to make the reaction go like this guy does. And this is something which we came up with in 2006. And this paper came out in Science, and it is now one of the more interesting discussions on how it's not exactly this, by the way. There are still some possibilities around here, but it is something like this. So that gives you the excitement of science here. We still didn't know what it is, but it took us all this time to get here. And we used the most sophisticated things available because facilities like what we have here. Okay, this is a very confusing picture, you may think, but this is a grand picture, which this is the same membrane, all the proteins. All that was found by other groups using different techniques, which also use X-rays. But what we found was the structure of this little tiny little thing which sits here, which I call the molecular engine of this whole system in a way, because it splits water. So it is water here, there's plenty of water. By the way, water, there's plenty of water everywhere, but it's also a very fascinating thing. And in Stanford, they are doing some groundbreaking, amazing work on the structure of water. And it's a lot more than what we think it is. It's not the simplest molecule. It is an extremely important, complicated molecule. And finding out how it works is fascinating, to say the least. So anyway, this molecule has a lot of water around it. It works with light. So you turn on the light, all these pigments, which are chlorophyll, which is what plants are made of mostly, largely, absorb the light, funnel it into the place where the reactions take place, and beautifully it takes water, splits it into oxygen and protons and electrons. This is hydrogen. So it takes water, splits it into oxygen and hydrogen. Isn't that what we're trying to do with environmentally friendly energy production? Isn't that what we have all been told is the way, one way to reduce all our energy problems? These things do exactly that. So the plants have already found a way to do it. We just have to figure out how to do it. And that's what our research is about, finding out how this reaction works. Okay, where are we going with this? We know what it looks like, but I just want to give you a feel. This is some of the instrumentation which is there in SSRL. It probably costs $1 million to put this thing together. It's all there. We can go and use it. Apply for time. It is reviewed by our peers for how good our proposal is, and get 1 week of time and do the experiments. Something like this is just mind-boggling. If a single person wanted to put it together, it wouldn't be possible. And this system works with light. So we use lasers, which we can bring here, and study the system. So it's really fun. Look, there's a little tiny laser sitting here. There are already enough instruments there. The little tiny laser shoots, and this is the spinach sample. And then you try to figure out how it goes through the reaction cycle. And one more thing is coming up now, the LCLS in Stanford, which is going to produce very, very short, intense pulses of X-rays. And now we have a tool not only for studying the static structure. Static means as it sits in one place. But we also have a way of finding out how to get snapshots. So we are making progress because of, again, increments in technology. Okay, the last part of my talk, we talked about evolution and how oxygen is produced from water by these little bugs. We talked about the manganese complex, the catalyst, which is there in the huge protein complex, which makes this reaction possible, and what its structure may be, and how it functions. And the third thing I already hinted, it already does the reaction which we want to do. It produces oxygen and hydrogen. If you combine hydrogen and oxygen, you get water. There is no pollution then. You get the energy you put in as light back when you burn oxygen. You're not making any CO2. And even if you make CO2, use CO2 to do things with hydrogen, you're still going to bring it back here. So I'll tell you about a little bit. So we talked about history of Earth evolution and its impact on chemistry, physics, biology, and the wonderful little catalyst, the molecular engine. It's all sitting in this plant leaf, every plant leaf. And I want to tell you a tiny bit about its relationship to the energy, the environment, and the economy. This is clearly an extremely important thing because we are, without doubt, doing some nasty things to our environment because of our way of using fuel. And we need to learn from this some lessons. Many of you have seen these slides, but every time I see them, it's mind-boggling. Okay, what are we doing? This is thousands of years. This is the present. This is 400,000 years. And this is carbon dioxide. Let's take one at a time, the black one. So carbon dioxide goes up and down because of various geological reasons. I'm not an expert on that. That's what happens. Okay, fine. Right now, it is at this point. It has never been there in 400,000 years. It's at 380 parts per million. It has never been that high. So something is going on here. And look at the red one, which is the red one on top of it, is the temperature of our Earth, mean temperature. That keeps pace with that. So we are doing something pretty bad. And one of the things we're doing pretty bad is this. We are multiplying like crazy. And that is not, the more people, we use more energy. It's not that, there is always this talk about certain people use a lot more energy, but this is much more so than the second part. If we had lesser number of people, we would be using lesser amount of energy. Okay, this is interesting. So this is where we are. We need to do something about it. Because once CO2 comes into the atmosphere, it takes a long time for it to dissipate. Hundreds of years. So to bring that thing down here is going to take us a long time if we can stop it there. We cannot stop it there at this point. That's taken for granted by all the experts. It's going to go up. But we need to slow down the process. And by the way, many of you have probably seen Al Gore's Inconvenient Truth. Remember, he goes up in a little lift up to show this point. This is the same point. He had to go up in a scale to see where it is. The same thing if you expand the scale. See, this is over now only 1,000 years. This is where the Industrial Revolution started, 1750. Both of them correlate extremely well with carbon dioxide and global temperatures. So along with Industrial Revolution, we are burning a lot more fossil fuels. What are we saying when we burn fossil fuels? You're burning plants, which is carbon, and you're producing carbon dioxide. And plants are not able to absorb all the carbon dioxide back. The cycle is disrupted. We need to interrupt the cycle. And in order to interrupt the cycle, we need to be able to produce fuel in a different way. This is the same thing I just told you about. One more thing. This is an interesting website to go to. It's the carbon footprint calculation from BP. BP, in spite of being a petroleum company, has very interesting facts on its website. And this graph, let's not go into too many details here. It just tells you the United States puts out about 20 tons of CO2 per person. China and India are here. France is on a flat line because it uses a lot of nuclear power. So there is a lesson there that in order to get rid of CO2, we need to use some other forms of, I'm not advocating anything, by the way. I'm just pointing out the facts. But we are up here, 20 tons. And the average US household uses 18 tons. Not uses, sorry, produces 18 tons. So I went to this website, and a typical Berkeley person has a small apartment, has a bicycle, but travels a lot, and it's 8 tons per year. But let's take my colleague, who is very, very energy conscious. She has 2 old cars and recycles everything, including a lunch pail, paper bag. And what do you think her footprint is? 20 tons a year. So we can't simply say 18 tons is too high. I'm not doing that. Even if you live very carefully, we do produce a lot. Okay, couple of interesting slides. This is not directly related to our research, but to just bring it back to the present day. This is the total power usage for various kinds of power. This is, of course, you can see gas, coal, oil, obviously are the highest. The reason they are the highest is because they are the cheapest. The lowest is renewable, which is solar, wind, whatever, and that is the highest cost. So this is a mirror image. We need to do something about that too. We need to increase this, renewable. It's a complicated slide. Don't read everything here. Just 2 things I want to point out here. We use a lot of energy, I shouldn't say energy. Energy and power, you have to be careful, but anyways, 13 terawatts. It's going to go up to 28 in 2050. It is going to go up. China, India, all other countries, United States. So what do we do about it? We really do need to use solar. Solar energy, as there's enough energy hitting the earth, enough energy to be able to use it. And how do we use it? Use it the way the plants do. The plants have shown us how to do it. Split water to hydrogen and oxygen, and combine them to produce energy. So this is a beautiful slide because this is how nature works. It uses photosynthesis to make starch and oxygen and then burns it up and makes it back again. So we have disrupted the cycle. We need to do it in a different way. And how is it being done around the world and in Berkeley and in Stanford? They're trying to do exactly that, learn from the leaf. We have a leaf, and this is the same system which I showed you in a different form. You can try to make an artificial membrane. On one end, you can oxidize water to oxygen. On the other end, you make hydrogen. Hydrogen has a bad rap because hydrogen is difficult to transport. Hydrogen is hard to compress, hard to make in dense form, and all those things are true. And how do you get around it? So you produce hydrogen and then use hydrogen to make something else, which is more like a fuel, in methanol. Same membrane system, which we learned from the plants. On one end, you make oxygen. The other end, you make hydrogen, which you use to make methanol. But you saw the common theme here? The common theme is you're always making oxygen from water. You need to know how to do that, which is what we're trying to do. That is important. However much what you do at the other end is not as difficult as learning how to do this chemically. And most recently, my last but one slide, so we're almost there. Our colleagues at MIT, Matthew Cannon and Daniel Nocera, published a paper in Science just this month, where they learned to split water. This is just a beaker of water. There is an electrode. You can do that. Anybody can do that. You've done it in high school. You put 2 electrodes, turn on a current, you split water. You see bubbles of oxygen. But these people learned how to do it efficiently. They learn to do it getting more energy than what you put in a way. And they were able to do it plentifully, which means they were able to do it at a very rapid rate. And we brought this system. This is a cobalt catalyst. You saw iron and manganese. Cobalt is right next to that in the periodic table. So in order to make a viable system, you need to get a common metal which is cheap. We brought this to Stanford here during the last week of July. And we actually collected spectra using the same kinds of techniques we use to study the biological system. So the same thing applies here also. How does this catalyst work? And it's very fascinating. If it works, it does. Can you see those bubbles? It's the same kind. You're passing current through it, and oxygen is coming through on the catalyst. Now it's a catalytic reaction. It's not simply electrolysis, which happens if you turn on a current. So it actually is doing the reaction in a very nice way. So it is splitting water. If you can split water cheaply, you can make the hydrogen on the other side, use it to make fuel. Okay, finally, this is the take-home slide. We talked about how oxygen and this complex made life possible as we know it. We talked about how it may work or it's put together. And we talked about how the same complex is coming back right now to help us save the environment and our energy problem. The same complex. And this is a nice comment from one of my colleagues. If a leaf can do it, we can do it too. And my final slide is a beautiful view from our lab in Berkeley. And we're eternally grateful to the Department of Energy and our granting agencies. And I thank you all for your attention.
Well, thank you for this fascinating talk, and we do have time for questions. I should also remind you that please, after the questions are over, please stay around. We have cookies and refreshments outside. But now I open the audience for questions.
I have two questions. Should I buy stock in manganese? And secondly, the result of a catalysis is protons, electrons plus oxygen. Do you combine the protons, electrons to produce neutral hydrogen?
Exactly. And the first question was, you asked why manganese? Is that what?
Yeah, no, should he buy stock in manganese?
There is manganese everywhere. It's one of the most plentiful elements next to iron. So, but I have the question reminds me of something which I forgot to tell you. Why manganese? Why did nature pick manganese? Because I don't know exact reason. Nobody knows the exact reason. But manganese chemistry is very different from the other elements. It is very versatile. It is willing to adapt itself to different kinds of structures. It's willing to adapt itself to being electron deficient or electron sufficient. And all those things are important in order to do this reaction, which, remember, we have not been able to do efficiently in a beaker. If you try to do it in a beaker, you will end up blowing it up or creating or putting in a lot of energy to do the reactions. That's right, because in the very beginning parts of life, 4 billion years ago, there was not that much photosynthesis going on. There was very little of it. So there was much more of the oxygen being produced. And sorry, I got the question. So there was not enough respiration because oxygen is burnt up by respiration. So oxygen could build up because of that.
So, but I think the question was, until very recently, was it a stable system of oxygen and CO2 and respiration? And I think the answer is, with these cycles, which you have seen also in temperature, it was pretty stable. So the amount of oxygen, actually there has been a period, maybe you can talk about.
You're talking about the 400,000 year cycle?
Yes.
I think that is actually well modeled using the most of these curves which we saw have been, scientists have put all the equations they could figure out into it and try to see whether it follows that. And it does follow that very well, except the last part. The last part is clearly something above and beyond, and you have to put in more things into your equations, and that is clearly what humans have done. So,
All the way in the back first.
So my question is, do all of the different photosynthetic, there's like C3 plants and C4 plants and cyanobacteria, do they all use the same manganese catalyst in the center of their protein?
absolutely.
Just one.
Just the same thing. The solar spectrum, the plants use the entire solar spectrum efficiently. Entire spectrum, which is very nice. There are spikes, of course, but they can absorb light in one part and send it to another part. So they do that. Yeah. The most efficient part is the red part, but they can absorb in the blue also. That's right.
How do you know that those are just one?
Very good question. That's what, because if you have 4 manganese atoms and you also had those units I showed you, try to put it together in any other way, you cannot. There are 4 manganese atoms and 3 of them are like this. How can you do that? You cannot do that. That's what the puzzle is about. You cannot physically put 4 manganese atoms together and also have 3 of them together like this.
Do you ever think of like a helix structure or different geometries in terms of?
That's right, yeah. That's what I showed you in the 2006. It looks, and it probably kind of, like, it needs to be in a certain way in order to bring in water, go out, bring in water, go. And by the way, it does it very, very, very fast, biologically speaking. It does it almost 1,000 times a second. It's almost like if somebody calculated the speed at which water has to go by, it's like a stream in order for it to do this reaction. And while it does that, it breaks apart also. After about a few million times, it breaks apart. It puts itself together. It repairs itself.
What about the idea that geometry, I found it interesting, the geometry is very similar to the geometry in a water molecule. You know, it's got the same.
That might be coincidental, but that kind of geometry is very commonly there in chemistry, that kind of 120-degree geometry.
Can you talk a little bit or explain, I mean, you've been using this equipment here at SLAC. Can you talk a little bit about the experiments and what you're actually doing and how you're getting the data out or anything?
I would love to do that for hours on end. I could do it in my sleep. Well, the important thing is you can tune the X-rays to the element you want. Okay, that's a very important thing. And you can use very, very intense X-rays because these systems are very dilute. There's very few atoms in a very large complex. So you need to be able to do that. And you also need to be able to do it without damaging the system. They're notoriously damage sensitive because X-rays damage things. We know that. So they're notoriously, so you need to take care of all those things, the garbage in, garbage out. So you need to make sure that you're actually looking at the real thing. That is, you're not damaging it. And the spectra you collect are many kinds of spectra. It's not just one kind. And you bring them all into play here. And that would take a little bit more to explain all those things. There is absorption experiments, emission experiments, and a whole slew of them like that.
If you ever come to a tour at SSRL, we can show you the equipment and actually the places, the hutches where these experiments take place. One here, and then we have the next one.
Joy.
Since the cyanobacteria evolved so early, has anybody tried one of those primordial soup experiments where you approximate what was in the ocean and in the air at the time and see what kind of
recreate the beginning of life?
I think it's a very interesting question. And what we have done, manganese is plentiful in myriad forms in minerals. So we went and looked at all these minerals. Obviously, these bugs picked up this complex either whole or one atom at a time. We don't know how we did it. It all had to happen in one bug and one little tiny thing somewhere. But we don't know if it pulled this complex intact or it took it one by one. Assuming it took it intact, so we went and looked at all the various structures possible. And many of the ideas we came up with is inspired by those structures. So, and actually, if you think about it again, what you talked about, if you have 4 manganese atoms, there are only a certain limited ways you can put them together.
There was one here. What's so special about spinach? Yeah, let me repeat. The question is, what is so special about spinach?
It's good for you.
Yeah.
And
Popeye.
we buy about 10 bunches of spinach, and we only take the greenest and the freshest and the nicest. We all become real connoisseurs of spinach. And the rest of the spinach goes home for making soup. That's one good reason. This is a question which a lot of people wonder, why spinach? Maybe one could ask, why E. coli to do biological research? You need a standard system which is commonly available across the world to all the scientists, so they can use the same species, the same kind, and easily available, easily grown. It grows in plentiful quantity, and you can get it fresh. Freshness is important because when you get wilted spinach or slightly old spinach, or my colleagues will tell you, I mean, they go to the specific store. Andronicos today, Berkeley, Mart somewhere else tomorrow, various things like that. But that is the reason spinach can be easily available everywhere. But people have used rice in Japan, peas, tobacco in where? Tobacco in
South
Cuba, various places.
And they can smoke the sample afterwards.
I saw recently a documentary about the evolution of the large reptiles about 300 million years ago. And according to that, the oxygen level had reached about 30% in the atmosphere. You show a maximum of our present concentration of oxygen on your curve. What happened between then and now, when the oxygen level was about 30%, which is credited with the evolution of large plants and food animals there?
There is this theory that there was a spike in oxygen. It's now leveled out at 20%. But there is some evidence that there actually was a spike of oxygen, went up to 30% and came down. And during that time, certain systems evolved, like the dragonfly, which is a foot long, and certain things. There is controversy about that spike, whether it really did occur or not. And again, I'm not an expert on that. But either way, it is fascinating that actually it did happen, and it came back down. And right now, it is at 20%. But there are those huge, monstrous things which did live during the time where that spike is supposed to have happened.
But it's a pretty important, an important result of photosynthesis, producing the food chain of the large plant. Anyway, thank you.
Right.
Let's have another question first.
One question. All the new equipment here at SLAC, I'm going to get it wrong, LSRC, or the new setup, what will that allow you to do that you can't with the
See, this
So
is
I'll repeat the question. So what is the new X-ray laser called LCLS? What that will help for the future of this type of research?
in a general way, every technological improvement has been enormously important for understanding systems which we have studied. And this is going to be an absolute quantum leap, this LCLS. There is nothing like that anywhere else in the world, which is the way SSRL was in the 1970s. And it's going to be much more than that, because it's going to be extremely short X-ray pulses, and you can follow things in real time. You can shoot your system with a laser of light, visible light, then it then shoot the short X-ray pulse, and see it evolve in time, all the way from the tiniest seconds, which are not relevant to biology but to chemistry, all the way to biology. And I think it's almost like we don't know what we can get from there. We're just waiting. Wow, let's see what we can get. That's my answer from a.
So you want to see not just how plants do it, you want to watch them do it.
Yeah. You want to be a warrior.
Okay, please.
Do you have any idea which atoms would be most likely to be in the active site that are actually doing it rather than the structure of the...
Absolutely, yeah. We have a lot of ideas. Lots of groups are working on it. And that is the nuts and bolts of the chemistry. And that's what is going on right now. We want to figure out.
You don't have a hint yet.
Yes.
We have lots of ideas, yes, and how it does it. And the way probably nature does it is in the most benign way, because it does it at room temperature. It does it at ambient pressure. Remember, if you want to do it industrially, for example, production of ammonia, fertilizers, we use enormous pressures and temperatures and use a catalyst. But nature does it in the most benign way. In order to do that, its chemistry is going to be also a little different. And that's what makes it interesting to figure it out. And it's a race now. And we are in it.
Okay, let's have one more question.
I was wondering about the protein framework that this catalytic reaction takes place. I presume that's the chlorophyll. Is there a lot of variation in the structure of the chlorophyll or the sequencing of the chlorophyll from species to species or from class to class of photosynthesizing organisms? Does it tell us anything about whether photosynthesis is evolved independently, maybe even several times during evolution of other organisms?
Very good question. There are two questions here. If you talk about photosynthesis itself, which is using light to make fuel for the plants, there are very many different kinds of ways it does it. There are many different pigments, many different, what should we call, photosystems, the one which does the reaction. And it's a fascinating topic because they're all tuned to their niche. One uses green light, red light, brown light, I mean not brown light, some other light, all kinds of light. And one grows at higher temperatures, lower temperatures, below the water, and they all have their little niches. And that is very fascinating subject in evolution and the branching of the trees. The other part, the oxygen evolution part, it is the same in all of them, what surrounds it, more or less. It found the right thing, it stuck to it. Not just the manganese, but the immediate surrounding is very similar.
Okay, so let's please all thank our speaker again. And please enjoy the refreshments. Vittal will also be here for some more time. And I hope to see you again in October.